Good morning from Traverse City, Michigan. We made it through the winter and we are on the upswing from the COVID. Speaking of the pandemic, I want to thank scientists like Dr. Fuller. It is their dedication that has taken us to finding protection and a new way for our community to function and move forward. Speaking of the pandemic, it's times like this that one must realize that there are different schools of thought when it comes to the science and looking for solutions. It's times like this that I really miss my father and would love to have been able to engage in conversation about solutions based upon his scientific wisdom. Enjoy the Saturday morning physics today. Our family is very proud to be sending this to you through the James Robert Walker Memorial. Enjoy and open your mind to all of the possibilities that we all have in front of us. Thank you.
Thank you, Diana, for those opening remarks. We're truly grateful for your family's ongoing support for Saturday Morning Physics and for sponsoring today's event. I'm Roy Clark, Professor of Physics and co-host of Saturday Morning Physics. It's my great pleasure and honor to welcome our speaker today, Dr. Evita Fuller, Professor of Microbiology and Immunology at the University of Michigan Medical School, who will present the James Robert Walker Memorial Lecture. Dr. Fuller is a renowned virologist who studies and teaches about human virus pathogens, including influenza, herpes simplex, HIV/AIDS, and more recently, COVID-19. She's a member of the U of M Africa Studies Center, where she developed an innovative public health program known as the Trusted Messenger Intervention, which partners with networks of religious and community leaders in Zambia and South Africa and sites in the USA to enable effective engagement of infectious disease prevention. And incidentally, that's where I met Dr. Fuller on a trip to Africa, which was really exciting. Dr. Fuller earned an undergraduate degree in biology and a PhD in microbiology and immunology at the University of North Carolina, Chapel Hill, and completed postdoctoral research in virology at the University of Chicago. Along with research grants from the National Institutes of Health and the National Science Foundation, she has received many awards, including fellowships from the Ford Foundation and the Fulbright Program. She is an inaugural alumna of the Ansbacher Women in Academic Medicine Leadership Program at the University of Michigan Medical School and is currently serving on the national committee that examines COVID-19 vaccines for approval with the Food and Drug Administration. Following our presentation, Dr. Fuller will be pleased to answer your questions, so we ask that you email those to us at this address, physics@umich.edu, and that address will be live streamed during her lecture. Now, without further ado, welcome again, Dr. Fuller. We're very much looking forward to your presentation.
Thank you, Roy. Thank you, Dr. Clark. Good morning. It is a joy to be here to share with you this morning on this Saturday morning physics series. I never thought I would be doing a seminar in physics, but thanks to the invitation from Dr. Clark, so it is. This morning, I also want to thank the Walker family for this lecture, sponsoring this lecture, and for all of you for coming out to see what it means from milkmaids to messenger RNA. So let's get started. What we're going to do is talk about pandemics, and then we're going to look at milkmaids and vaccine discovery, and then we're going to talk about a very popular topic right now, COVID-19 vaccines, and then we'll talk about your questions and answers, hopefully. So, what are emerging microbes, epidemics, and pandemics? We need to define some terms so that we all are on the same page. Emergent diseases are infectious diseases that are caused by a microbe that first appeared or increased in incidence in an area or in a new location. An epidemic is affecting at the same time a large number of persons in a locality. A pandemic is when there are multiple epidemics. Infection is present throughout an entire country or throughout an entire continent or region or globally. And then we have resurging microbes, which are like the measles virus, where it was shut down, but then it resurges in a particular area due to lack of vaccine uptake or immunity. What factors promote microbe emergence, epidemics, and pandemics? Well, we have a bunch of them. And I'll just point out a few that might be of interest to us at this point. So enhanced human-animal interactions, microbial evolution, the change in the microbe, the widespread trade for food and other items, the population changes where people are migrating from the rural areas to the cities. And then when emergencies happen and we have breakdown of infrastructure, changes in human behavior, how easily we travel now from one part of the country to another or from one continent to another. And then, of course, war and unrest. All of these things affect microbes. COVID-19 is not the first, as you know. We have a history of pandemics since the time humankind existed. And this slide actually shows you some of those all the way back from the plague, the Black Death as it was called, which essentially changed society. It was when the feudal society was changed from having lords and serfs and others to where there were so few people that people got to work in different areas. Then there was the smallpox pandemic, which we're going to talk a little bit about. And so coming down, we also find things like the 1918 flu and yellow fever. And guess what? This is COVID-19 on this timeline. And this ball keeps growing as more people are affected by coronavirus 19 or coronavirus, SARS coronavirus 2, which is the causative agent of the COVID-19 disease. And so if you look in art history, you'll find pictures of things like the Black Death represented here. If we compare these pandemics that have happened over the history of humankind based on the amount of people that died, so the death toll as a result of a microbial infection, we see by far that the Black Death around 1300 was the greatest one in terms of mortality. And then we have smallpox, which we're going to talk about today. And here we have COVID-19, which was at some point over here as a very tiny ball, but as more people around the globe die from coronavirus infection, this continues to grow. So this was the amount as of March 17th, 2021. And again, you see art pictures depicting what was going on in real life. So how does this work? These microbes are transmitted in various ways. And so I just want to make sure you understand some can be done by direct touch of persons or objects. There are things like vectors, which can be insects or even other viruses, which we'll talk about. Some can be airborne, and that's what coronavirus or COVID virus is. And then some are through food and water intake, like hepatitis A virus. So the mode of transmission matters. And so I want to do a demo for you. When we have a population where a virus transmits to a population or a group of people that are not immune, that have never seen that before, or have no vaccine and have never seen that virus, something amazing happens. And this is a physics setup. As I said, I never thought I'd be doing a lecture in a applied physics program, but I'm so delighted. So let's see what happens. If each of these represents a person, but they've never seen smallpox or coronavirus, which emerged as a new virus, or any other microbe that's new to that population, When that microbe begins to reproduce in that not immune population, this is what happens. Nothing. Well, let's try it again. Wow. And that first one may have been one that wasn't adapted to humans yet, but the second one, which now is a better human pathogen, just triggered a huge response in that whole not immunized population. And so we get lots of death like this when a population has never seen that microbe and there is no vaccine, no natural immunity or vaccination. What do we do to help take care of this? Well, we can, if it's an airborne microbe like SARS coronavirus, we can do these things that we've been told to do for the last year, where we keep our distance, we stay home if we don't need to be home. This is actually my first day back in a building on campus. It feels a little strange. We can limit the size of gatherings. We can correctly wear a mask. I've had my mask on the whole time I've been here. We can wash our hands and not touch commonly touched items. And when we feel symptoms or think we might have been exposed, we can get tested so there can be contact tracing and isolation until we know whether or not we're infected. And then there's been lots of scientific research on things like vaccines and anti-therapies. Here's an example of some of the masking or protection that was used with the Black Death. So you see that the doctors covered themselves up. They had masks and hoods, which allowed them not to breathe in much of what was going on. Again, this is depicted from artists from that time. And this is the modern masking up, right? This is what we do. So this is someone getting a coronavirus test or COVID-19 test, but you see the physicians or health personnel are all masked up, much like this person in ancient times. And then, of course, we can wear a mask to be sure that we're keeping away from things that allow the virus to go into our mouth or nose. So let's stop for a minute and talk about 3 or at least 2 emerged human viral pathogens that have had a huge impact on the world. One is smallpox, which is caused by pox viruses. Another is orthomyxoviruses, which is like the influenza virus that we get a vaccine to every year. And then the third is coronaviruses, which is what causes COVID-19. Pox viruses are large DNA viruses. They're pretty much one of the biggest viruses there are. And it caused smallpox, a devastating disease that I'm going to talk about. Influenza is an RNA virus. And I have here some demos. This shows you the double-stranded DNA, which is what the smallpox virus DNA would look like. And then if I just had one strand of this would be an RNA. It would be moved to be part of a molecule called RNA with just one strain, sort of like this coil. And that would be what the influenza virus is made of and what the coronavirus is made of. We're going to talk about coronaviruses in a minute. With orthomyxoviruses, we get a flu shot every year because this virus genome can change. And we're very concerned as to whether it will change or we'll get recombination such that we get bird flu or swine flu that comes into humans. You may remember the swine flu epidemic some years ago when a porcine or a pig virus acclimated to humans. All right, so let's talk about smallpox. Smallpox has been around since, I think, humankind. This is an example of the mummy of Ramses V that was discovered from 1143 BC. And you see that if you look closely, his skin has these scars which are reminiscent of what happens when someone gets infected with smallpox. It actually leaves scars from these large lesions that occur on the skin of the body. So smallpox killed approximately 500 people in the 20th century. Survivors were badly scarred by the pockmarks or blinded by corneal ulcerations. And you see this woman is blind because the virus actually caused lesions in the eye and destroyed the cornea. And we already talked about Ramses V. There was a process in China called variolation, which would try to help to decrease the amount of infection, but sometimes it transmitted smallpox. And then in the 1500s, during the, quote, discovery of America by Europeans, even though there were people already here, some of those Europeans brought smallpox on objects like blankets and gave them to the Aztecs, who were a non-immune population. And what happened, just as you saw with the demo, they were wiped out. Over 90% of the people died from exposure to a new pathogen, smallpox. And then we can look in art also to see examples of viruses that have been, or artwork where people have depicted what has happened. So now, what happened in the smallpox outbreak, and how do milkmaids affect this? Edward Jenner, who was a scientist of that time, was variolated. He was used that early process from China and almost died as a kid. He noticed, however, a sharp scientist, that milkmaids who had been infected with cowpox, of course, milkmaids milk cows, right? So the cow virus, cowpox, when they got those lesions on their arms and hands, seemed to be protected from smallpox. He noticed that the milkmaid's facial complexion was clear compared to the pox scars of people who got smallpox. Do you remember the Mother Goose tales where they would talk about the fair, fair milk maiden or fair, fair maiden? That's what they're talking about. They had smooth skin because they didn't get smallpox because they were exposed to cowpox. This is what cowpox looks like. So someone who's milking a cow would get this on their arms and hands. And so Jenner, using the scientific method, observed that milkmaids do not get smallpox. They are exposed to cowpox. He hypothesized that the material in cowpox lesions contacted by the milkmaids protected them from smallpox. And so he conducted an experiment. And this is a statement by Thomas Jefferson, who wrote to Jenner in 1806, "Future generations will know by history only that the loathsome smallpox existed, and you have been extirpated, and has been extirpated by you." I think that's how you say that word, extirpated? Okay. So here's the experiment. On May 4th, 1796, Jenner used cowpox infection material from the hand of Sarah Nahmes to successfully vaccinate young 8-year-old James Phelps, and this is captured in this print. About 6 weeks later, on July 1st, he challenged the boy by deliberately inoculating him with smallpox material. And guess what? James Phelps did not get smallpox. Voila, immunization. So this was the first vaccination. We couldn't do those experiments today because he obviously had no human clearance or IRB approval to do such an experiment. But we found out that giving smallpox to someone who was not immune gave them, or giving cowpox to someone who was not immune gave them protection against smallpox. And so the term vaccination, which we mean when we give someone a vaccine, comes from the Latin word for cow that was coined by Louis Pasteur. And this is just a print of people around 1905 in France coming to get vaccines to smallpox, a free vaccine. So you see the baby is being inoculated, the woman's being inoculated. This man is extracting the cowpox lesion to get the vaccine to expose the people. And so we collected cowpox for this vaccine to then expose to people intentionally. And then by 1876, we had noticed that somehow there had been a shift, and instead of the cowpox virus, we were using something called vaccinia. By the 1960s, we were trying to vaccinate the whole world, and this was a little difficult because it was hard to do in places where there was tropical places. And so heat-stable, freeze-dried portions of the vaccine were developed. By 1967, after that, there were only about 15 million cases worldwide of smallpox, something that had killed millions and millions. By 1977, the last known case of smallpox was reported in Somalia. And in 1980, the World Health Organization declared that smallpox had been eradicated. So this is what someone who looked like who was sick with smallpox. Very, very not good. And this, some of you may have this scar on your arm as a result of the smallpox vaccine. I know I do, and I see Roy shaking his head. We're telling our age when we say we've got the smallpox scar. All right, so that was the first case of vaccination to help eradicate smallpox. And there are other conditions that were required, which I won't go into. But the point is that vaccines were developed. So what are vaccines and what did they do? Vaccines prime the immune system so that when we actually see the microbe, we do not get the actual disease. And so we have some very good vaccines. Smallpox, as we just talked about, yellow fever, measles, mumps, rubella, the MMR vaccine that we all get as children, polio, chickenpox, pneumococcus, human papillomavirus, a relatively new one, and even in the last couple of years, Ebola, and now we have COVID-19 vaccines. All of them are looked at for how safe they are and how well they stop the disease that the virus or microbe causes. There's some things you have to do to have a successful vaccine. You have to have the vaccine before infection. So you have to get the vaccine before you see the microbe. We have to determine what form to use. We have to determine when to give it to the host. Do we do it just once or twice? Is there a booster? We have to decide where to inoculate. Is it safe and how to store it? We have to measure the response to the microbe and to the vaccine. And then, as you know, we have to distribute it so people can have access to it. But there are some great benefits to vaccines. There's high uptake. If there's high uptake, then we have something called herd immunity. And that's where enough people in the population are vaccinated and immune, protected from the microbe, such that when the microbe comes, it can't reproduce. And those few people who are not vaccinated are protected by the ones who are. Think of a herd of cattle surrounding a newborn to protect it. In order to do that, though, you have to have high uptake. Does it prevent disease and does it prevent microbial transmission? These are all factors that we think about with vaccines. So the vaccines that have been developed recently for COVID-19 are vaccines that fit all of these criteria. They are part of the virus known as the spike, which is shown here. So these are the projections of the virus, and that is encoded by one of the sections of RNA. And these vaccines contain the RNA encoding the spike. When we have influenza vaccines, if we can get a 70% effective flu vaccine, we are like, it works great. So let's talk about these vaccines and the COVID pandemic. Very briefly, you know this, so I'm going to go through this pretty fast because you've lived through this. We have lived through this history. The vaccine, or the virus that causes COVID-19 is called SARS coronavirus 2. It was originally called novel coronavirus because it was a new coronavirus, one we had never seen before. It transferred from animals, which is called a zoonotic transfer, into humans. And we have lots of these all the time. Swine flu, SARS, MERS. And so there are lots of viruses in the coronavirus family, over 100. And we think that they reside in bats. And then when animals come in contact with the bats and humans come in contact with the animals, the viruses get transferred to us. So that's what we see here. We have about 5 or 6 different kinds of coronaviruses before the SARS-2 appeared. And so when it appeared in November of last year, it caused the disease that was ultimately termed coronavirus infectious disease 2019. And it began to spread around the world in a pandemic. So on March 11th, 2020, almost a year ago today, the World Health Organization declared that there was a worldwide epidemic or pandemic of COVID-19. And so you can see here some of the most highly affected countries, of which the US and some countries in Europe are. So between that time in November and now, over 2.7 million cases or 2.7 million deaths have come from COVID-19, and over 121 million reported cases, which means it's probably even much higher. So this clearly qualifies as a world pandemic. The whole world has been sat down by SARS coronavirus 2, a new virus introduced into the human population that transmits through the air. What has it done? So here we can compare the pandemic in the US. And so we see that of the countries represented here, Japan, Canada, Europe, or continents, the US, we are by far having the highest number of cases. And we see here that it even disproportionately affects some groups of people. So this is data from Chicago early on in May. And we see that if we look at cases across Latinx, Asian, white, and African Americans, it's African Americans that had the highest number of cases, in part because of the work we do, the places we live, and in part because of underlying conditions. And then the deaths follow with the high cases. And so this is illustrated in another way here from a few states. The population is on the left. The percent of deaths of COVID-19 that were African American is on the right. So for example, here in Michigan, we are 14% of the population as African Americans, yet by May of 2020, we were 41% of the deaths from COVID-19. So these things have to be addressed, and there are people, including our governor's office, who's working on this. So what do we do about COVID-19? How do we stop this demo that we showed from occurring with COVID-19? Besides the protections like using masks and staying home and keeping our distance, do we have vaccines? So yes, I'm happy to say we do have vaccines. And so vaccines can be delivered orally or on the arm, but the key is what's in the vial. So let's look at this. Dr. Clark mentioned that I'm on the FDA advisory panel to do reviews of vaccines. And so we now have a bunch of these that are in the pipeline in what's called phase 3 clinical trials. All of these vaccines went through the normal process of what happens to show that something works and it's effective. And because it was an emerging situation, we were in the middle of a pandemic, it was done a little bit quicker, but with all the same steps and with all the criteria reached. So right now we have approval of Pfizer, BioNTech, and Moderna, which are both messenger RNA viruses, and I'll explain that in a minute. And we recently, in February, had approval of Johnson & Johnson, which is an adenovirus vector vaccine. And so with all of them, we had to look at efficacy and safety. And these are just some videos that help explain what an RNA vaccine is. And I'm going to show you one here, hoping that this will help.
Process of creating copies by
Let me start it over.
Researchers have found that the COVID-19 virus begins the process of creating copies by attaching its spikes to a host cell. Those spikes offer a target for vaccines. With the right preparations, our immune system can utilize this vulnerability. A vaccine is used to arm our immune system. It contains messenger RNA, which can direct cells to make specific proteins. The messenger RNA tells our cells to produce COVID spikes. These harmless spikes give our immune system the information needed to quickly detect a coronavirus infection and produce the appropriate antibodies. The new antibodies respond once COVID has entered the body. They attach themselves to the spikes on the COVID virus, rendering the spikes useless, protecting our cells and keeping the virus from reproducing. mRNA vaccines have been studied rigorously and are a safe and effective way to defend ourselves against COVID-19 infections.
So this is an example of an adenovirus with the spikes. And it's the spike protein that actually is used to get immunity to SARS coronavirus 2. What do I mean? So what is in the messenger RNA vaccines? Let's just review. So here we want what's going into the arm to be something that will protect us from SARS coronavirus 2. It's not the spike protein, it's not the genetic material of the virus, it's just a small bit of the RNA that encodes the spike protein that is put into a fat droplet, a little bit of fat like you have when you boil chicken or when you cook with oil. These droplets carry the RNA into the cell. They're put into a vial and dehydrated, and then the messenger RNA in this material is rehydrated at the site. When it goes into the person's arm, the fat droplet with the spike protein encoding RNA gets into cells, is taken up by cells. Because it looks like material the cell would normally use, the cell actually begins to make spike protein from the code. And that material, that protein spike, goes to our cells, and now our body says, aha, this cell has something on it that does not look like me, that's foreign, and it mounts this wonderful immune system. Our bodies are wonderfully and fearfully made, such that whenever we see something foreign, it actually recognizes it and begins to destroy it. And so because the vaccine puts in the messenger RNA that makes the spike, we mount an immune response that will destroy anything that looks like the spike the next time it is seen, when we actually see the real virus. All right, so that's what these messenger RNA vaccines are. Were they tested? Yes, they went through all the clinical research trials. So there's laboratory work that my colleagues around the university do. There's a phase 1 study, that's a safety study that takes about 6 to 12 months. There's a phase 2 that takes anywhere from 6 months to 2 years normally, where we're looking more so at effectiveness in people who have the condition. And then there's the big phase 3 studies that have thousands and thousands of people, typically over 5 to 10 years. But because we were in an emergency situation, companies and researchers worked together to develop the messenger RNA technology. And by the way, it's not new technology. It had been used for cancer delivery. It had been used with other items. It was just putting it together with the messenger RNA, the right piece of the COVID-19 virus, the right lipid particles to come together to produce a vaccine that would be tried. And so there were over 30,000 people in each of the trials for Pfizer and Moderna, the 2 messenger RNA vaccines where you get in 2 doses. And there were even a greater number of people in the Johnson & Johnson vaccine, which is a slightly different one that I'll talk about, because that's only a single dose, so it only needed to be in people for a shorter amount of time. So we have emergency use authorization for 3 vaccines, where there are many others in the pipeline waiting to get the results to see if they work effectively. So these vaccines that are available now have gone all through the process that shows that they are effective in stopping disease symptoms and that they are safe. The Johnson & Johnson vaccine is the newest one. So instead of going into the cell as a fat droplet here, it used a carrier virus to deliver the spike gene that ultimately does the same thing, which is to make the COVID-19 virus spike on our own cells. All right, so let me just summarize. I know that's a lot, but let me summarize. We have 3 vaccines that are now emergency use authorized and available to people for COVID-19. 2 of them are messenger RNA viruses. They have the coronavirus spike binding protein that's made in our own cells. It takes 2 injections. For Pfizer, it's 21 days apart. For Moderna, it's 28 days apart. But they block up to 95% of the disease symptoms that people get if they get exposed to the virus. So out of 100 people, only 5 who get the vaccine in the studies would have any symptoms when they get both doses. The Johnson & Johnson is a virus vector that actually uses another virus to deliver that spike protein, and it's up to from 67 to 85% effective in stopping the disease from coronavirus infection with only 1 dose. And it also works for the current variants that are circulating. We don't know how long the immunity lasts, and there are side effects, and you've heard that. But the side effects, which are symptoms of the disease, or actually symptoms of our immune system mounting a response, only last for a couple of days, and they're well recognized. So if there are side effects, and some people have them, some people don't, you can report these to the CDC at the vSafe, and it's very important to do that. Or if there's something really serious that you think you want to talk about, there are other numbers to call. So just to review then, vaccines are a way to protect against virus. The vaccines that we have are putting in the spike gene from coronavirus into either a fat cell or either into another virus. And this virus, by the way, is replication deficient. So the RNA would go inside such that when that virus goes into the cell, it now allows that cell to make the coronavirus spike protein so that our immune system mounts it against the coronavirus. And so let's demonstrate this one more way. This would be DNA, which has the code. The coronavirus is made of RNA, so it would be only one strand of this, and then a small snippet of that. So let's say this is the RNA. A small piece of that encodes the spike protein. That is what's in those little bubbles that goes into our cells to make spike protein so that our immune system can now recognize it and destroy it. Why do we want to do this? Because COVID-19 is a very serious virus. Let's just quickly review. It is a respiratory spread virus in aerosols. When we breathe it in, or somebody who doesn't even know they're infected has virus being produced, and they talk or sing or sneeze or exercise, these droplets come out and they fall on nearby surfaces and they remain suspended in the air, such that anyone within 6 feet or closer without a mask will breathe them in. The virus will infect their nasal passages, travel down the throat, the windpipe, the cells here into the lungs, and infect the very small cells along the way, including the alveoli cells where we exchange oxygen. And this is a picture of a lung now that has mucus and is so clogged that you can't get air through. That allows fluid to build up, and so you end up getting pneumonia or severe acute respiratory syndrome. People say it feels like there's an elephant sitting on their chest. They can't breathe. When that happens, body organs begin to fail because of lack of oxygen. So there can be no symptoms. There can be mild, moderate, or severe symptoms that range from simple things like pink eye and loss of taste and smell to very serious things like neurological issues, strokes, pneumonia, blood clots, heart disease, kidneys failure, immune system failure, blood clots in other places. And in the most serious cases, people are hospitalized and they have to be given oxygen. And if possible, they survive, but many people don't. As we know, we've had over 500,000 deaths in the US alone. So these vaccines block COVID-19 disease and death. All three of them do this. These are the symptoms that may appear with the disease. And for some of these, we see them when we get the vaccine because our immune system is responding just like we respond when we see the virus. COVID-19 affects people in different ways, from no to mild symptoms to severe illness to death. But the good news is that the vaccines prevent the disease symptoms and they prevent illness and death. And so therefore, we need to take everything we can in the toolkit to stop COVID-19 pandemic. Mass distancing and not gathering still must be used whether we get a vaccine or not. And so in summary, I hope I've helped you understand the importance of milkmaids in first discovery of how vaccines work with cowpox and smallpox, and then how we have used that same theory and developed vaccines that will stop COVID-19 disease. And what we're doing is not presenting the virus, but presenting the spike protein on our own cells, either taken in on a fat droplet that carries the code, the messenger RNA, into our cells, or a defective virus that takes it in and allows that to be produced, such that our own cells produce the COVID spike, just the spike, so our wonderful immune system can mount a response, either B cells for antibodies or T cells for protection. And then when we actually see the virus, we have a memory that allows us to say, I've seen that before, let me destroy it. And so that primed immune system from the vaccine comes in and destroys any coronavirus that we see. And so this is how we can get out of the pandemic. We must have high uptake in order to get the required protection to protect our population. And so as my last thing, let me show you what happens when you get at least 2 out of every 3 people in a population vaccinated. So again, here are people, and now instead of no immunity, we have 2 out of 3 people that are either immunized, hopefully, or they might have survived a natural infection. But we know that natural infection can also lead to severe disease, long hauler syndromes, and other things. So let's see if it works. Okay, so maybe that was the mutant virus. Let's try another one. 2 people out of 3 are immunized. There's no pandemic. Look at that. Can I try one more? 2 or 3 out of 4 people are immunized here. Let's try throwing it over this side and see what happens. Wow. What happens when people in a population are immune to a virus? There is no pandemic. So I hope that you have enjoyed this Saturday morning physics time, and the takeaways are, pandemics are a part of adaptable nature. It's just how the world is made, that nature can change to adapt to the environment. And so sometimes the contact between human, animals, and environment lead to a new microbe going into a new population. That's what happened with SARS coronavirus 2. For COVID-19, you must make an informed decision, but for my money, at 95% to 65% effective in preventing COVID disease, even for a little while, a COVID-19 vaccine is a wonderful place to start to help us manage, all of us manage COVID-19 pandemic. And then lastly, whether you get a vaccine sooner or later, you must continue to use the things that we know work. And that's the masking, the distancing, the not gathering, the hand washing. Even as the pandemic goes down, as the number of cases go down, we don't want to let up on the gas pedal until we're safely out of this, until there's very few, less than 3% positive cases of COVID-19 tests coming back. So I thank you for your time and your attention on this wonderful Saturday morning. I would like to acknowledge the people I work with in research, what we call the Trust Messenger team. I want to thank Dr. Clark for his invitation, who I met through the STEM initiative of the African Studies Center. Bethany Moore, who I used a few of her slides on smallpox, who's also chair of the microbiology and immunology department. Monica Wood, who helped set up these beautiful herd immunity model systems. And then my spouse and family, who as always support me. So I would say to you, this was fun with physics. Thanks for engaging, and now it's your turn to ask questions. You're muted, Roy. We can't hear you. That was actually fun. Imagine that you can demonstrate what happens in a pandemic with a physics concept. I learned something today. Pretty cool. So this is always the question when we get on technology, particularly Zoom these days. It's like, okay, I'm talking, but nobody can hear me. So how do I unmute? We've got a bunch of questions, and I'm just going to start with the one at the top. Oh, there he comes. Great. Yeah. Super.
Yeah, so I'm sorry about that. So thank you very much, Dr. Fuller, for a very fascinating and informative presentation. I want to start this question and answer session by recognizing the outstanding work of Monica Wood, the manager of our lecture demonstration lab, who put those demonstrations together. We love those elegant mousetrap simulations that she put together, illustrating virus contagion and how to immunize for herd immunity. So thank you, Monica.
Yes, absolutely.
So we have a lot of interesting questions to get to, and let me start with the question that came in just now, and this is to do with the mechanism of administering the vaccine. And the question is there a problem if I get one dose of, say, the Moderna vaccine, both mRNA vaccines, but let's say I get one dose of Moderna vaccine, and then weeks later I get a dose of the other mRNA vaccine, the Pfizer vaccine. Is that what are the issues surrounding that kind of situation?
So first of all, it would be great that the person is getting vaccinated, because that's what it's going to take to get us out of this pandemic. Second of all, the recommendation is that if you get Moderna for the first injection, that you get Moderna for the second injection. Or if you get Pfizer for the first one, you get Pfizer for the second one. And that's because there are proprietary items, not a lot, in the lipid nanoparticles, and there's different amounts of messenger RNA. So it just makes sense that you are getting the same one. As far as what will happen, I don't think we know, because I don't think those trials were ever done, or there was no need to. Each company did its own. So I would imagine, however, that you'd still get a pretty good immune response, but I wouldn't advise someone to do that because we just don't know. But both of them allow expression of messenger RNA into spike protein that then actually allows the immune system to mount a defense against the spike.
Thank you for explaining that. There's some related questions about safety of vaccines, and one of them is a little bit technical, at least for me, and
Okay,
I think you'll be able to handle this better. So the mRNA sequence of the Pfizer vaccine is reportedly 4,284 nucleotides long, and is one of several that researchers decided on to develop the vaccine. And the question is, what guided the researchers at Pfizer to use such a sequence, and how are these different than those developed in the 1990s when certain animal tests were done, which revealed some issues related to that. Could you address that rather technical issue?
Yeah, I'll give it a try. So the sequences, the 4,284 sequences, I'm impressed that the person knows this. These are to the spike protein that are allow it to be locked in what's called the pre-fusion state. And so that just simply means that the spike protein, and I'll just use this analogy, if my hand is the virus, is the coronavirus, my fist, with all 5 digits. When we make a vaccine using the messenger RNA technology, or even with the Janssen vaccine that uses an adenovirus vector, a defective virus with the spike gene, we're not putting in the whole virus. We're putting in just the gene or the messenger RNA that makes this thumb, which I say is the spike, which attaches the virus to cells. And so that those nucleotides that the person asked about would be encoding the full fusion protein or the full spike protein. And it's actually in a manner that if you imagine the fusion protein at some point might be kind of tucked in, or when it actually allows the virus to fuse with the cell, it might extend. So it's just that Pfizer and Moderna chose a form of the spike fusion protein, the code for the fusion protein that keeps it in a certain conformation. And so that's what's in that sequence. And they chose that because they looked at a number of different sequences and picked the one that was most protective. That is, that made the body's immune response occur in a way that most protected the initial animal studies that they did. And those studies have been going on for a while. We just adapted it for SARS coronavirus 2 when that sequence was known. And then how is that different from the vaccines developed in the 1990s? This was to SARS and MERS and other things when mice died from inflammation. Well, with the messenger RNA, it turns out that if you put just messenger RNA into a cell, it actually is a very high immunogen, and it actually, if it survives, if you transfect that DNA or that RNA right into cells, you get lots of expression, and that's where you got inflammation. And so by putting it into a lipid bilayer or lipid nanoparticle, choosing the very best immunogen or the best RNA that can make the best protein, the studies early on, which had actually been done before coronavirus, SARS came along, or SARS coronavirus came along, actually showed that you get a good immune response, but you don't overdo it to get that inflammation that was so detrimental early on in the studies back before 2010. Mm-hmm. So we had the best of many worlds. We had people who were studying coronavirus. We had people who were studying messenger RNA. We had people who were studying lipid particles. And because we were now facing what could have been a pandemic, which did turn out to be a pandemic, those researchers came together, shared their information, worked cooperatively, and scientists did this amazing thing of creating these messenger RNA vaccines. And the fact that there are two of them that work essentially from independent places really confirms the beauty of the science from two different companies.
Thank you. I must say I'm really impressed by the technical background of our audience and in asking some of these questions. Another one that's related is to do with encapsulation of the mRNA protein in lipids and how that's done, and using nanoparticles, and how are those nanoparticles chosen, and are they different than the ones that are used for producing human erythropoietin, HEPO? I'm not sure I pronounced that correctly.
Right, you got it. Human erythropoietin, yes. So again, we had much of this technology around, using it for different things. So the nanoparticles, which is that lipid or that fat droplet that I showed in the video, is a technology that has been developed over the last 25 years or so to take things into the body and deliver different agents. So I mentioned cancer therapeutic agents have been delivered by lipid nanoparticles, or in this case, human erythropoietin has been delivered. And so those particles were used to take items that you wanted to be around for a little while, like the drug in cancer treatment or in hormone treatment. For the vaccines, we don't want it around a long time. We just want the messenger RNA to get in and get into cells, to trigger that cell for a short time to make enough coronavirus spike protein that it can be presented to the immune system. So the technology works beautifully for delivering a short-term primer of the immune system, which is what the messenger RNA of the spike does, as opposed to much more detailed stability had to be done for delivering the drugs to cancer cells, or particularly for delivering hormone treatment through nanoparticles. So this was a lot simpler in many ways with what we're trying to do because we only needed it there for a short term. That messenger RNA is quickly destroyed in the cell. It doesn't integrate into the chromosome or the genome. We won't have, 5 arms and 2 heads, as some of the conspiracy theories say, because the messenger RNA is just a code that tells the cells to make the protein and then it's destroyed. Next question.
And I'm guessing that the use of nanoparticles is not as much of a concern. My own area is actually on nanoscience and using nanoparticles for different applications. And we're always concerned about them in the environment and what kind of problems they can cause, for example, liver damage. But since they're encapsulated in this way that you described, presumably, and for a short time application, presumably that's not a major concern for their use in this application to vaccines.
Yeah, and one thing I would like to mention is also the side effects that we see when some people see side effects, some don't. Apparently, the older you are, the fewer side effects folks seem to see. But like my niece who works in a residential home is in her 30s, and she had a headache the next day. So I think that the side effects are, one, our immune system is seeing something that's not us and it's revving up. Even when some people get the influenza vaccine, they have side effects. They feel a little bit feverish and body aches. That's not bad. That means the immune response is gearing up and saying, I see something that's not me and I'm going to attack it. So that's what we're feeling. And it might also be a little bit of response to the nanoparticles immediately. That's, I think, why people are asked to stay 15 minutes or more right after the vaccination to make sure that those particles, the nanoparticles, that, the injection doesn't cause a problem. But the side effects are really a sign that our immune system is working, and those are short-lived. They're usually gone within 2 to 3 days. There may be some fatigue or tiredness the next week, but that also goes away as we're mounting. Imagine your body is making a response to that foreign protein. So it's taken a lot of energy to do that. And that's why we feel a little bit tired.
Thank you. So another question is on a lot of people's minds right now is to do with the efficacy of vaccines. Is, for example, 96% efficacy better than 65%, and in what ways? So that's a question that perhaps needs some explaining. Could you
Sure.
say a few words about that?
I can do the best I can. So, as human beings, we always want to compare things, and we want the best of things. And really, all 3 of these vaccines are really excellent vaccines. As I mentioned in the seminar, we get annual vaccines to influenza. And some years, those vaccines are great. And some years, they just don't seem to be effective against the circulating influenza virus. And that's because it's a guess as to what's going to be circulating the next year. Those vaccines get made a year from the previous year's circulating strains predicting the next year's. So in a year when we have a really, really good flu vaccine, you know what, Roy, it's about 65 to 70% effective. We think that's a really great flu vaccine. And so all of these COVID-19 vaccines are at least as good as that. And they go all the way up to, as we've seen, 94 to 95% efficacy against COVID-19. And let me explain what they were evaluated against is their ability to stop any symptoms of coronavirus infection or coronavirus COVID-19 disease. So fevers, chills. In the study, people who were in those trials, if they got any symptoms on a list of COVID-19 symptoms, then they were considered to they were considered a case. So fever, headache, body ache, chills, difficulty in breathing, loss of taste or smell, all of these, if you got those, then you were considered a case. And the data showed that people who got the vaccine, if they got any symptoms at all, that they got very mild symptoms. But most of those people, I'll give you an example of how the data looked. If you had 100 people or 200 people who now reported to the investigators that they had some sort of symptom that I just described, they were put into the cases. And so when you got up to a certain number of cases, based on the number that you had to have a certain power of effectiveness, you could break the code. When I say break the code, neither the researcher nor the person in the trial knew whether they got the placebo, which was the salt or sugar water with no vaccine, or whether they got the vaccine that contained the messenger RNA or the adenovirus carrying the spike. And so those who, when the code was broken, and let's say we had 100 cases, we actually had more than that, but let's say if we had 100 cases, and we look at all these people who had some sort of symptom reported, those who had the most severe symptoms were always those who did not get the vaccine. They got the placebo. So 95 or 94 out of 100 people who got symptoms had not gotten the vaccine. And only 5 of the people out of the 100 who had symptoms got the vaccine. And even more so, the 5 that got the vaccine always had milder symptoms. So they might have had, maybe they lost their taste, or maybe they felt feverish for a day, but they never got the really severe symptoms that are associated with COVID-19. So that's how we get to the efficacy. So imagine even that out of 100, 65 people did not get the vaccine, and they got more severe COVID symptoms. And what's that leave? 30 left, 35 left. Those people had very, very mild symptoms if they got any at all. So the data from even the Johnson & Johnson is very protective against severe symptoms, which means that it doesn't matter which of these 3 you get, that it protects against the thing that we want to happen most, which is keeping us away from severe symptoms at home, or even having to go to the hospital, or even, heaven forbid, people dying from COVID-19. So all 3 of these work beautifully well. And even the Johnson & Johnson with 1 dose works against many of the variants that are circulating. Johnson & Johnson tested its vaccine in Brazil, South Africa, and the US, and there were variants that we know are circulating in those populations. So the Johnson & Johnson vaccine also worked reasonably well against the variants, which is very, very good news.
Mm-hmm. Yeah. Indeed.
So
yeah.
all 3 of them are good vaccines. All 3 of them are, I would take any one of them.
Yeah, that is super good news. And it's a really good point that these vaccines, having access to several different kinds, and all of which are very effective, is a real godsend.
And may I just say one more thing with the Johnson & Johnson? The nice thing about it is that it doesn't require the same sort of storage. So that means that it can be used anywhere in the world much more easily. And that means that we can go into difficult areas or tropical areas or places where even the refrigeration and freezing with Moderna isn't necessary. So we've really got a nice toolkit of vaccines that can be used in multiple places, because unless every place is safe, then no place is safe, because this is a respiratory transmitted virus.
Yeah, so that kind of brings up a point about our work, that we're both involved with some work in Africa, and specifically in healthcare, delivery of healthcare in situations where it's very difficult and there's very little, if any, electricity, for example, to operate a refrigerator.
Right.
So we've been looking into solar powered refrigerators, which don't operate at such low temperatures, but they're fine for a vaccine like Johnson & Johnson. And that's a really important thing to have in a difficult environment like rural Africa.
And that was, as you may have noted in the talk, why smallpox took a long time to be eradicated, because we had to get into the pockets where we couldn't take a vaccine so easily. And so once it was dehydrated and it could be resuspended, that's when we could take it into areas where refrigeration wasn't available. So this is really important that the Pfizer vaccine can be used in places like Michigan, where we have the minus 80 centigrade freezers to store it. Moderna can be used in communities, also at U of M and other places, but the Johnson & Johnson vaccine can go places that those other two cannot, and still protects well against symptoms, disease symptoms, and death. And so we're really grateful for that technology.
Yeah, it's really good. So another question is, how long does it take to develop a vaccine? This is a very interesting question because, we didn't start developing vaccines in March 2020. Tell us a little bit about that,
Right.
if you could.
Right. And that question also, underlying question is, can we trust these vaccines since they were developed so quickly, right? We all heard about Operation Warp Speed. Well, the great thing is that, as I mentioned, I hope I mentioned that these vaccines were developed because the technologies for the various areas, the lipid particles, people who had been studying coronavirus spikes because of MERS and SARS, and the ability to stabilize messenger RNA, those things were being studied, not because there was a coronavirus pandemic or a worldwide one, but because scientists are interested in things. So when the first news of this new novel coronavirus came up, people at the NIH and other places who were studying the various parts, actually, once the sequence was known for SARS coronavirus 2, then it was realized that we could use the technology we've been developing for messenger RNA vaccines for other things, and now switch it to the sequence for this new coronavirus, do some modeling, which allows us to know exactly what we need to put into that, what messenger RNA we need to put into the nanoparticle to get the protein we want that's going to elicit highest neutralizing antibodies. So in other words, people came together. I make the analogy that if you were working on a quilt and I was working on a quilt and somebody else was working on a quilt, we each had our square and we were planning to complete our own quilt. Instead, we come together and put our squares together and keep doing what we're doing, and we can get to a quilt much faster. And that's exactly what happened. The cooperation between scientists who were doing different pieces of this puzzle came together to work together because we were in an emergency. So normally it takes somewhere between 5 to 10 years to take a vaccine or a product through the various steps I showed you, from the preclinical laboratory studies to phase 1, phase 2, phase 3. With COVID-19, the preclinical ones have been done. And so starting in January or maybe February or maybe March at the latest, the phase 1, 2, and 3 studies began to happen in a cooperative way, such that we could get to this in a very unusual time without skipping any of the steps. The only thing that has not been done, and this is because it's not possible, is to know what's going to happen 5 years or 10 years, because we haven't had these vaccines that long. But for me, I believe that, as with most vaccines, that they're safe, and they, once they're in the body, the immune system, which is amazing, will handle what needs to happen, and we can get rid of this disease. We still don't know what happens once they're there for 5 years, but with most vaccines, that is not an issue at all. And we're hoping that will be the case. Those studies are underway, but we don't know the results because we haven't had them in people for years yet.
Yeah, still learning, and we'll be learning for a while. That's right.
We will be learning, that's right. But one thing we do know is COVID-19 is a very dangerous virus, right? So we know that, and the vaccines protect against the symptoms. Even if you don't go to the hospital, long hauler syndrome with COVID-19 is a big issue for people who get infected. And that completely changes people's lives as well. There's so much that happens that we don't understand with the COVID-19 virus.
Here's another question related to the storage of vaccines. And it's to do with, so you've got one vaccine that requires minus 80 degrees storage, the Pfizer vaccine, whereas another mRNA RNA vaccine like Moderna can be stored in a normal kind of freezer, like temperature maybe -20. What is the science behind that? Could you say a few words about that?
Yeah, I really must say I don't know the details of why one is -80 and one is -20, and one can be fine in a refrigerator for 3 months. And that is company proprietary information that we weren't given. But I imagine it's what's in the lipid and what other buffers are there to stabilize those lipids. But we weren't told that. But I'm just glad that we know exactly the conditions for each one, and that there's lots of quality control from the time the vaccines are manufactured at the manufacturing site through the time that they're distributed to the location and even at the location. So I don't know exactly what about these besides that it's the stabilizing buffers. And I know for the Moderna that there are very few other things in there besides the lipids. There is some SDS, which is a type of detergent that's in there. And some people are allergic to that. That's also found in many other vaccines. And so for those people, you may have heard of some people getting an anaphylactic response at the site. And that's probably not to the messenger RNA lipid particles, but to that SDS. So those are people who, if they get a flu vaccine or measles, mumps, rubella vaccine, they get a response. And what we do is we want them to take their epinephrine pen with them to the injection. They ask to stay 30 minutes to make sure there's no immediate response. And it's that solution that helps stabilize the nanoparticles that they're probably responding to. But most people have no problem with that.
Mm-hmm. Yeah, so here's another interesting question. This is to do with the fairly recently a Nobel Prize winning development to do with genetic manipulation called CRISPR. And what's the relationship between CRISPR and the technology used to develop these vaccines?
Wow, we are getting...
Yeah, we're getting into the grass here.
We're getting into the tree leaves, right? So CRISPR is just a way of doing some fast amount of analyzing a lot of human genomes. And I don't think that CRISPR was used very much for this because the sequence was already known for. I mean, we knew specifically what we wanted to put into these viruses based on the work that had been done with previous coronavirus. And so there are lots of high throughput DNA and RNA studies that can be done these days. Technology is amazing. But for this, the researchers who did the modeling, and that's been done over years to know the structure, the molecular structure of the coronavirus spike, that was the major thing. And once you have the sequence of the novel coronavirus and you have the protein structure of any coronavirus spike, you can actually take that sequence and do computer modeling to see how the sequence of the new virus stacks up with the sequence that you knew in the protein structure, and then do the adjustments to know what to put into animals to see what brings out the best neutralizing immune response. And so that didn't require some of the high throughput technologies that we have. If you're looking for the gene, if you had a new virus and you didn't have a clue as to what would be immunogenic, then you would need those high throughput technologies. But we knew the spike protein of coronavirus was the target that we wanted to use. So let's not ask such technical questions because that's hard to explain. But no, ask whatever you see. That's fine. I'll do my best, or I'll say I don't know.
Yeah, I'm grateful for that, Dr. Fuller. So another question is, do some cells in the body accept lipid nanoparticle, I guess those are nanoparticles encapsulated in lipids, and produce antibodies better than other cells?
Yeah, so the question is what actually happens when those lipid nanoparticles get in our body? And I'll be honest, I really don't know, but I'll just say that what I imagine ultimately happens, it goes into some cells of the body, those cells make spike, and then those cells now are detected, and particularly when it goes into any of our antigen presenting cells like macrophages or others, who's designed to say, oh, this is something different than in my body. The spike protein is expressed, and then those cells now are activators of our immune system. And so that could be an immune cell, or it could be any other kind of cell. The good news is that the messenger RNA and the lipid particles are broken down really quickly, so they don't hang around. That we're not trying to deliver a drug to stay there for days or a gene to keep expressing. We just want to get a little bit of protein made such that it triggers our immune response. And that's what happens in whatever cells that those lipid particles get taken up into. And it's really pretty cool technology.
Yeah, it is.
I see a question.
I guess the last part of that question was to do with, is the vaccine tuned to be active on certain receptor cells? And I'm guessing it is. That's been kind of the refining of this kind of vaccine.
I'm not sure I understand that question, but let's. I see one that I would like to answer, if that's okay.
Yeah, please go ahead.
Why does it take more than one virus particle to create an infection? Am I jumping ahead of you in the list? So actually, we're calling that the how much virus does it take for someone to get infected? And we know that whenever someone who is infected, whether they're having symptoms or not, if they're producing SARS coronavirus 2, they're producing lots of virus. I didn't show any pictures in the lecture, but if you see cells that are producing SARS, they're producing lots and lots of virus particles, hundreds from one infected cell. And so I don't know how many particles it takes to infect a person, but I know that those droplets that we breathe in when someone who's putting out virus or who's infected and breathing out virus, just not in a cough, but just talking like I'm talking, and you are nearby without a mask and they don't have a mask, then we breathe in lots of particles. And that's certainly enough to infect cells in our nasal passages. And once it's infected, the infection goes really very efficiently. And that's one thing we also know about these virus variants, like the Great Britain or the B117 variant or the one from South Africa or Brazil. At least the Great Britain one seems to be much more easily transmitted. Now, what does that mean? Does that mean that it takes fewer particles to get into our body to have us be infected? Or does it mean that the few ones that get in can reproduce faster to make more virus? Or can they invade the immune system quicker? We don't know. We just know that that variant seems to be able to be more easily transmitted between persons. So I'll give this example. If you are let's say you're in the supermarket, right? And you are shopping in an aisle and you see someone who maybe even has on a mask, but I have a mask right here. Let me see if I can pull this up. Instead of having the mask on properly, they have their mask. So this will be the proper wear, but they have it on like this, right? In the supermarket. And so the air they're breathing out is going into the air. And if you are, let's say you're trying to figure out which kind of cereal you want to buy, and you want to stand there a few minutes and, read the different labels. So with the regular coronavirus, if you're there for 15 minutes and you inhale some of that air, you might get infected. Or 30 minutes, or let's say 15 minutes. With the variants, you only have to be there about half that time. So 5-10 minutes breathing in the variants, and there's enough put into your body that you get infected. Or say you're out with friends at a restaurant, right? And you're gonna enjoy an evening out, and you're there for an hour. Well, if someone's producing the new viruses, the variants, then more people will get infected from the air they breathe out than if it were the regular virus. And so these new variants are much more transmissible. But the thing we want to do is to prevent virus replication in anybody, because the more we give the virus an opportunity to reproduce, the more opportunity there are for new variants to arise. And whereas these have not escaped the 3 vaccines that are out there, we don't know what the next variant will be like. And so it's so important, so important that all of us do everything we can to prevent more virus replication. And there are at least two ways to do that. One is to follow the preventions we already have, the masking, the correct masking, so covering both the mouth, the mouth and the nose, making sure the sides and the bottom are tight, and keeping it on the whole time you're around other people. And then keeping that distance, that 6 feet distance, such that any droplets that fall are not inhaled. And if you don't have to be around other people, don't be around other people. Washing your hands to make sure anything you touch. So those are the preventions we already had that will help us drop the number of infections. And then the new one, the new kid on the block would be the vaccines that we have. 3 vaccines that can reduce the level of disease. And hopefully we're still looking at this, whether they produce virus express, which is why people who get vaccines still have to use the mask and the distancing. But if we do those things, we can drop the amount of circulating virus, drop the number of people that get infected, and thus reduce the chance of illness as well as the chance of virus variants. And so that's so important. Even if you get a vaccine, you still need to do those things because we don't know yet that the infection levels, the person who has a vaccine is not making virus. We hope they're not, and it seems to be less virus, but we're not sure yet. So I wouldn't want it, if I'm vaccinated, to be around someone and the virus I'm making, even though I'm not sick, will inoculate somebody else.
Yeah, so really good point. So another question is for the vaccine, if somebody has been vaccinated correctly and 100%, with both doses, if it's a 2-dose vaccine, have there been any cases of serious illness that has occurred or infection, I should say, even though the person has been fully vaccinated?
Right. Yeah. So in the studies, people who got the vaccine, there was not illness, which was really the exciting data. And what I like, and I think you may know that with the initial Pfizer review, I was one of the 4 people that said no for that first vote, because first of all, I thought there was going to be a second vote and we could ask some of the questions about things that were not yet shared. There was not, so it still was approved. But the point is that I got a lot of comfort from learning about the Moderna vaccine the next week, where those questions were allowed to be asked. We changed the format such that FDA changed the format such that questions about long-term duration and the level of infection and how many people were in the study that were of a different ethnicity or race could be asked. But the thing I get great comfort from, Roy, is that now that we've actually rolled out the vaccines, so the trials were done in 30,000 people and 40,000 people for Pfizer, 30 for Moderna, but now it's gone into millions of people in the US even. And what we're seeing with the actual rollout in terms of what you ask about symptoms after you get the vaccine for side effects as well as disease symptoms, we're seeing pretty much what was seen in the clinical trials. So there's nothing that's popped up that we're aware of that's so different than was in the clinical trials. And I know there's information circulating that says there were people who died from those clinical trials. Well, yeah, there were people in the clinical trials who died, but there were no more people who got the vaccine that died than people who got the placebo that died. So if you get together 30,000 people or 40,000 people and you follow them for 6 months, chances are somebody is going to pass. Is that right? Because yes, we are human beings, but there's no correlation with or without with the vaccine. And that's what we see also with the rollout of the vaccine in real life. And that gives me lots of comfort. That's why that V-Safe information that you get when you get the vaccine is so important to log in on your phone so you can get those messages, and you'll be checked on every day for a week to see whether you're having what your side effects are like, and then for several weeks, once a week, to see how you're doing. That's how the data is being collected for the rollout of the vaccines. So we know what's really happening in reality. And if somebody gets really something that they don't understand, they can always call their health provider or call the CDC VAERS number, which should also be given to you when you get your vaccine.
Another question.
Did I answer your question? I think I did. I hope I did.
I think you did. So another question is about the AstraZeneca vaccine in the US. Is it possible that a 1/2 dose is more effective, or that the booster shot is not needed? I don't know if you can answer that, but
yeah,
please
well, I
give it a
can't
shot.
answer for AstraZeneca because we haven't reviewed that, and I really haven't looked at their data. But I can answer that for Moderna and Pfizer BioNTech. And so the question is, would we be able to get more people vaccinated if we just did one dose to spread that out further than keeping doses such that we can give each person their booster or their second injection? And so the answer to that is the same thing that NIH is saying, is that because the studies that got people up to 94, 95% effective in reducing coronavirus symptoms were done with 2 doses, then that's the better way to do it. We know what happens there. If we start giving people 1 dose, which does provide some protection, and somewhere around 40 to 60% protection, that's better than nothing, but it would be better to follow the protocol. So even if you can't get your second injection within the prescribed 21 days or the prescribed 28 days for Moderna, get it whenever you can. So maybe you can't get back to the site in 21 days. So maybe it's 40 days before you get there. Well, go back and get that second injection, because that is going to take your already primed immune system, introduce the messenger RNA that encodes the spike for just a moment to now boost your immune system to even a higher level. And we believe that protection will last longer as well as is more effective. So the two injections seem to be the best protection. That's what the clinical trials showed, and that's what we want to stick with. It's like if you do an experiment in your lab, Roy, you don't want to go and change something. You don't want to change things once you now get the assay working. You want to stick with what worked out, the best conditions you found. You don't want to go change it in the middle of the rest of the experiment, because then your results may be different and you won't know why. So that's why we're recommending that that both injections are done, even if it is a little bit later.
Great. We're coming up on the near hour shortly in a couple of minutes, but I would like, we've had a lot of interesting questions and we haven't got to all the questions, but I'm sure that Dr. Fuller would be happy to have a look at whatever questions we didn't get to and respond by email. So, but let me finish up with a kind of personal question, and that is, how did you get into science, Avita, and what does it mean to you doing research of this kind?
Wow. So thank you for asking. So I didn't, some people say I always knew I wanted to be a scientist from the first day I did such and such. I don't have one of those moments. I grew up on a farm with my father and mother and uncles and grandmother, and just was surrounded by nature, and I think was always curious. I was always that kid that says, well, why this and why that? And, they would explain something. I go, well, why? And so I think that's really the temperament of what we do as scientists, which is to ask questions or observe things and ask questions like Jenner did. He noticed that the milkmaids didn't seem to have the pox skin. And why is it that milkmaids don't get smallpox? And that led him to the hypothesis that ultimately led to him doing the amazing test with the 8-year-old boy that showed that when you get cowpox, exposed to cowpox, that does something to your body to prevent smallpox. So I think that curiosity is what I enjoy about being a scientist, that you can explore things, that nature, which is so amazing, so incredibly it works, right? And it's also adaptable. That's how these microbes get from the virus, from an animal virus into humans. So I didn't have one of those moments. It was in college when I had to decide between being a biology major and a journalism major that I decided maybe I could make a more reliable living being a biologist than writing, because people might not like what I wrote. And then I went on to study and do a paper on the Legionnaire's disease and got hooked on science inquiry and went on to grad school. So it wasn't one of those aha moments from a kid that I knew, but just a love for exploring things and learning. And that's what I still have. I just hope that we can learn together and do what we need to do to stop this pandemic and learn the lessons we need to learn to be able to handle the next one better, because there will be more. But we can do this.
Yeah. Thank you for those words. And that's very inspirational. So we're up on the noon hour. That really concludes our program for today. But I want to finish by thanking you again, Dr. Fuller, for a super informative presentation and question and answer discussion that you provided. And I want to say to everyone, please make sure to get your COVID-19 vaccination when it's your turn. A special appreciation also to the Walker family for sponsoring today's James Robert Walker Memorial Lecture, and to all of our loyal supporters of Saturday Morning Physics. I want to thank Carl Cole, our video and his video production staff at Michigan Media, and our communications director, Carl Raybuck, for keeping us all on track with this series of Saturday Morning Physics presentations this semester and in previous semesters. 2 weeks from now, on April 3rd, we'll have our final Saturday Morning Physics presentation of the winter semester, given by two of our illustrious graduate students, Ariana Bueno and Ryan Hubbard, who will be presenting some of their research being done in applied physics. And we look forward to seeing you all then.
Thank you for this opportunity. It was fun.
Thank you.